Welcome to 16.2. We are talking about the process of DNA replication. And if we remember at the end of the last set of notes, we talked about how Watson and Crick had discovered the structure of DNA, and they said that they noticed that the way that it's made could lend itself to understanding how it is copied. And scientists believe that there was 3 different ways in which DNA could be copied. One was the conservative approach, where the DNA would split in half, and then it would be copied, and then you would have you'd have a complete original strand, and then it would be a completely brand new strand. The dispersive copy was that it was sort of broken down into segments and then copied that way, and then mixed and matched so that you had 2 strands like this, half and half, like that. And then the semi-conservative approach, where the DNA strands were split into 2 and it was copied, and so each half of the strand would be brand new. So each new strand of DNA would be semi-conserved, half old and half new. And a very elegant, one of the most beautiful experiments ever completed was done by Michelson and Stahl. And what they did was they radioactively labeled the nitrogen in some DNA and in some Escherichia coli or E. coli, and they centrifuged it. So there's light DNA and there's going to be heavy DNA. The N15, which is the radioactive label, is going to be the heavy DNA. And so when they centrifuged it, it separated out. So the heavy DNA was at the bottom and the light DNA was at the top. And so they took the heavy DNA, and this is what they studied. They took the heavy DNA, and so we have the heavy DNAs shown here in brown, and then it was allowed to copy again in the E. coli, but it used regular light nitrogens so that they could track how much of the nitrogen, or how much of the light nitrogen was being used in each strand of DNA. So when the DNA was copied again, they found that the DNA, when it was put into a centrifuge, it didn't separate out some light, some heavy, because if it did, that would be the conservative approach. You'd have entire strands of light DNA and entire strands of heavy DNA. So they didn't see that. They found that all of the DNA was mixed. It was half heavy, half light. It was sort of a medium weight. And so that could either be semi-conservative or that could be dispersive. So here, remember, semi-conservative has half new strand, and the dispersive is kind of bits and pieces put together. So then they let the Escherichia coli divide again, or to copy the DNA and pass it on. And what they found was that there was some DNA that was completely light, and some of the DNA was that middle half light, half heavy. And so that couldn't be the dispersive, because the dispersive would have been, once again, still half light, half heavy. So this gave way to the understanding that the DNA was semi-conservative, because there's now, after a second round of replication, there's going to be new strands of DNA that are fully light nitrogen, and then there's going to be a semi-conserved strand here that still has half of the heavy nitrogen. And so that is the Michelson-Stahl experiment, which was pretty amazing. Please note that here is a website that I stole all these pictures from, and it's a great breakdown of the Michelson-Stahl experiment. So if you want a different explanation, please feel free to check that out. So our 46 chromosomes have 6 billion nucleotide pairs, which is crazy. So if it was expressed in this, if their A's, T's, C's, and G's were expressed in the size of the print in your textbook, it would take 1,400 of your biology textbooks to express your genome, which is a lot. So however, a single cell can copy it all within a couple of hours. So your DNA replication process takes just a couple of hours with errors about 1 in 10 billion. Sometimes the more I learn about life, the more I don't understand how it doesn't go wrong more often. So DNA replication. Let's start talking about a little bit of the vocab. It's going to start, let me grab a draw tool. It's going to start at the origin of replication, and that should make sense. So here is our origin of replication. This is a ring of DNA, so that tells us that it is a bacteria DNA. So here is our origin of replication. And that's where it's going to begin, and then it's going to work outwards in both directions. And so it's going to make this replication bubble, which is this big right here, this big thing right here. And the replication bubble is going to extend, so right where let me show you, it's a better picture here. The replication fork. Oh, it won't let me change without the draw tool. So right here is the replication fork. It is where the helicase is opening it up. And so here is your replication bubble. It's extending on both sides. So let me go back to the last page here. So in bacteria, the DNA exists in a ring form. So there's only a single origin of replication, and the bubble just gets bigger and bigger until it meets on the other side, and you have two separate strands of DNA. In eukaryotic cells, there are hundreds and thousands of replication bubbles per chromosome, because there's a lot of DNA, and so it's going to occur in both directions as well. So your Y-shaped regions right here, we said these replication forks, these are created by helicase. Helicase is going to untwist and separate the DNA. So it's going to go in, it's going to open it up, and then it's going to make the template available for the other enzymes to read. Now remember that your two strands of DNA are attracted to one another, so when it opens, there's going to be a tendency for it to close. And what we have here, you can see these little bubbles here. These are single-strand binding proteins, or SSBPs. They act as like little door stops to keep the DNA from closing back in on itself. So it keeps it separated so that the polymerases or the other enzymes can grab on and do work before it closes. Now, if you could picture, if I had a piece of thread, or not a piece of thread, a piece of yarn, and if I were to try and pull that piece of yarn apart, and because it's all twisted together, it would create strain in the front. It creates sort of it would sort of get all caught up, and that is what DNA would do because you are opening up the DNA and it's all twisted. So topoisomerase is going to cut and relieve the strain, and it's going to rejoin the DNA strands back together. Pretty incredible. So the topoisomerase relieves this strain that would be caused by the DNA opening. And so enzymes, remember this, think about this. Enzymes cannot synthesize a brand new molecule of DNA. They can only add on to an already existing chain. So that means if we go back to our cell theory, cells have to come from pre-existing cells. Life has to come from life. And interesting thought that the original molecule of life was thought to be an RNA molecule. And so all of life on Earth is an extension of that RNA molecule. It has been copied and copied and copied. And new life or new molecules or new DNA strands aren't just synthesized. It is replicated from pre-existing strands. So it is pretty cool that we are all related in that way. And once the template strand is available, an enzyme called primase will read the parent strand and lay down a strand of nucleotides. So right here, so primase, so helicase has opened this up at the replication fork, and we've got the SSBPs. And now primase is going to come in and lay down these orange pieces, or the primer. And now we'll do this, or the primase does this because DNA polymerase has to grab onto the DNA at these very specific spots where there is primer. And so primer is a single-stranded molecule. It is technically RNA. So here it is in the orange. And it's going to be laid down in the 5' to 3' direction. So it can get really confusing when you start trying to figure out which direction things are moving. So remember, so this is how I remember it, and you can always work backwards from here. Remember that you can only build onto the 3' end. I just think about whether it's DNA polymerase or it's primer, but if it's starting here, it's going to read from a 3' to 5' end. I'm not talking about reading, I'm talking about building. It can only build onto the 3' end. It can only add on to the 3' end. We're not talking about reading, we're talking about building. So replication only builds in a 5' to 3' direction. It can only add on to the 3' direction. If you can remember that, you can figure everything else, you can work backwards. So replication, or DNA polymerase, or any polymerase, can only add on to the 3' end. It can only build onto the 3' end. I hope that helps. You may not get it now, but you will once you start trying to figure out the directions of things. So there are several DNA polymerases. In eukaryotic cells, there are more than 11. And of course, they were named in the order in which they were discovered and not in their order of, what roles they play. So in bacteria, 2 appear to be the major players. So we have DNA polymerase 3 and DNA polymerase 1. So DNA polymerase 3 is the big player in replication. So DNA polymerase, what it does is it attaches on the primer. So here's the primer, the end of the primer. So it finds the end of the primer and it attaches on to that 3' end. It's going to keep adding on to that 3' end. So DNA polymerase 3 needs to find that 3' end of the primer and add on and keep adding to the 3' end. So in bacteria, the rate is about 500 bases per second, whereas in eukaryotic cells, it's about 50 bases per second, which is staggering. Each nucleotide being added to the growing strand consists of a sugar attached to a base. So we've seen this before. We've seen ATP, which is 3 phosphates, sugar, and a base. The difference between ATP and DATP. Let me just step back. So DATP is going to become the nucleotide that is added on to the DNA. So this is going to be, so ATP is a nucleotide, as is DATP. And let's look at ATP. We have 3 phosphates, a sugar, a ribose sugar, and a base. Now let's look at the molecule that is also a nucleotide that's going to be added on to your DNA. We call it a DATP. The difference here is that it is a deoxyribose. And which is going to our deoxyribonucleic acid, so deoxyribose. And it still has 3 phosphates right here. And then the nitrogenous base, those are going to be your A's, your T's, your C's, and your G's. So these are going to be variable, A's, T's, C's, G's. If it's RNA, U, but we're not there yet. And so this right here, the DATP, is going to be added by the DNA polymerase 3. So DNA polymerization, or the building up of DNA, is going to take this DATP, so here's our deoxyribose, here's our 3 phosphates, here's our nitrogenous base, right here, it's a thymine. So as DATP is added, 2 of the phosphates are going to be hydrolyzed, they're going to be removed, and we call these 2 phosphates a pyrophosphate. This pyrophosphate is going to be, removed, and that's going to give energy to the process of replication. And then this pyrophosphate is going to break down into 2 phosphates, individual phosphates, which is going to continue to add energy to the process of replication. So this is an exergonic reaction, which helps to fuel the process of replication. So during elongation of replication, DNA polymerase 3 is going to remain at the replication fork. So here is our replication fork, and this is our replication bubble right here. So DNA polymerase 3 is going to add nucleotides to the 3' end, always building on the 3' end. So here, as they are replaced, they need to be sort of glued down. And so that is the job of DNA ligase. Ligase is going to go in after DNA polymerase 1 replaces the primer, and they're going to glue in or sort of create that sugar phosphate backbone and going to form those bonds, and that is going to make your strand of DNA. Ooh, let me move this up. Here we go. So proofreading and repairs. DNA polymerase is remarkable. The initial base pairing process has been found to create an error rate of 1 in a million. The final product, though, has an error rate of 1 in 100 billion. So after the polymerases have finished the growing strand, the mismatch repair machinery, so these are more enzymes, are going to begin to replace, delete, repair DNA. And so when genes for these enzymes are faulty, so the enzymes that are part of the mismatch repair machinery, if those enzymes are faulty, it can cause a buildup of genes being read or defects in your genes, which can lead to cancer, and means that they can accumulate faster than normal, or errors in your genes can accumulate faster than normal. So after replication is complete, there are hundreds of enzymes constantly on the lookout, keep making sure that the DNA remains intact, because it is always under attack by things like X-rays, free radicals. So oxygen can be a carcinogenic, and spontaneous chemical changes. So cytosines can actually just break down or decompose into uracil. And so when that happens, they need to be, I'm covered again. They need to be fixed so that the DNA can be read correctly. So when an error is identified, an enzyme called a nuclease will remove the segment. So here, this green blob is the nuclease. It's going to remove the segment. And then the nucleotide excision repair system, which consists of a polymerase and a ligase, is going to go in and replace the missing nucleotides. And so this is going to happen constantly, not just during replication, but all the time in your DNA, because your DNA is constantly under attack, which is why we hide it safe in the nucleus. But even in the nucleus, there are still things that can get in there. And there's a disorder called Xeroderma Pigmentosum. It is an inherited defect in the nucleotide excision repair system. So we said that if those enzymes don't work well, that it can lead to a buildup of defects in your genetic material. And so in this individual, they don't have a nucleotide excision repair, and so therefore they are hypersensitive to UV light. So anytime UV rays strike and attack their DNA and break it down, there's nothing to fix those changes. And so this is an indicator of how often those enzymes are working for us and fixing repairs, because if this person can't do it, this is what we would look like if we didn't have it. And so this often results in skin cancer. And without sun protection, children that have this disease can develop skin cancer by the age of 10. So while mistakes are rare, they do happen. And some mutations, most mutations are silent. They're not even expressed. Most of them that are expressed are going to be harmful, but very few are beneficial. And the ones that are beneficial tend to be passed on. So if it helps an organism to survive, if it makes life better, easier to get energy, easier to reproduce, it's going to be passed on, or it can be passed on. And this is part of what causes the diversity of life. It was the only thing causing genetic diversity prior to processes like sexual reproduction. There is another video here. I'm going to add all of these videos to the description later on. So this is an interesting video about how mistakes in DNA replication led to snakes having venom, which allowed snakes to be more successful. So the ends of DNA continuously get shorter, at least in eukaryotic DNA, not in bacterial DNA, because they have circular genes. They have circular DNA. So in eukaryotic DNA, the end right here is going to have a primer. And so when the polymerase tries to attach on and to repair so the DNA polymerase can attach on and work to this segment, it cannot add on to beyond this side over here, because there's no 3' end for it to grab onto, the primer 3' end. So at the every time your DNA is replicated, it's going to get a little bit shorter and a little bit shorter. And this is already considered. So to protect the genome from erosion, there are telomeres at the end. So the very ends of your DNA have these things called telomeres, and they're just repeating segments so it'll just be a repeating segment, for example, like TTA, GGG, TTA, GGG, TTA, GGG. And they're repeated 100 to 1,000 times. This will allow for about 60 rounds of replication before genes start becoming eroded and having staggered ends. Staggered ends in a strand of DNA usually indicate a double break, which is a bad thing. And this would often trigger cell death. That means that the DNA strand is broken and it can't be repaired, and it would signal apoptosis or cell death. And so the telomeres prevent this from happening because they don't have genes on them, and so this won't signal the enzymes to come in and fix it and try, or to tell it to go into apoptosis. It's sort of in the don't worry about it zone. And so finally here, normal shortening of telomeres protect against organisms from cancer by limiting the number of divisions a cell can go through. However, there are some cells that have figured, so some cancerous cells have figured out how to fix those, or how to add on to those end segments. And so therefore they can continuously replicate. And these are lines of cells that, an example of this would be Henrietta Lacks's cancer cells. This was the first immortal line of cancer cells, or immortal line of cells. So this is not normal. However, it is a growing field of study because we can only imagine what that could do for us. I hope that helps, and please let me know if you have any questions.